Pretty thighs, shawty, she a dime. Demon girl, evil eyes, she be telling lies.
So your research lab, and then you mentioned you've developed a suite of highly efficient ACRA programs for the alignment of NGS sequences to large genomes and for the assembly of sequences from the RNA sequence experiments. I mean, I think the three programs are bow tie, top hat, and cufflinks. So can you explain a bit more about these programs and how you came up with all these weird names?
Oh, all right. Well, that's a series of programs that have been, bow tie is still the state of the art. Top hat was replaced by a program called HiSat, and cufflinks by a program called string tie, written by other colleagues of mine. And but basically, the way this all started was around 2008. A new technology came along. In 2007, there was this new sequencing method came along. It's called next generation sequencing, or now NGS. And sometimes we now more correctly call it second generation sequencing because there's now another generation that's appeared. And it was far more efficient and far faster, far cheaper than previous sequencing. So suddenly people realized they could do all sorts of things. DNA sequencing became so cheap, they could do all sorts of things they couldn't do before. And one thing that several different groups kind of simultaneously invented in 2008 was a method for sequencing the RNA that's being transcribed from DNA in cells in the process of making genes. So if you want to find genes, which is something I've been doing for a long time, you can use these methods that we were developing earlier where you're just trying to look at the DNA. But you could also you'll do better if you can say, let me find the parts of DNA that are getting copied into RNA, because those are definitely genes, or at least the genes have to go through that step. So if I can find the parts in the human genome, and again, it's only about 2% of the genome that gets turned into these protein coding genes. So this will give me a huge heads up, and it will let you do other things too. You can then, as an immediate sort of side effect, you can also measure how much of the gene is being produced in a cell.